the structure of DNA. The studies of the DNA began in 1868. By the 1940s, it was known that DNA is made up of linear polymers called nucleotides. A nucleotide consists of a pentose sugar, phosphoric acid and nitrogen base. Transformation of life science from biochemistry and agriculture to medicine and genetics was happened in 1953. In 1962, James Watson, Francis Crick and Maurice Wilkins jointly received the Nobel Prize in Physiology or Medicine for their 1953 determination of the structure of deoxyribonucleic acid. Please see the figure. The new era of molecular biology began in 1953 when they reported the discovery of the structure of DNA that genes are made of. Wilkins' colleague Rosalind Franklin, who died of cancer at the age of 37, could not be honored because the Nobel Prize can be awarded only to the living. They discovered that DNA consists of two long chains that form a double-stranded helix. Maurice Wilkins and Rosalind Franklin also contributed to this discovery by X-ray diffraction photographs of fibers of DNA. The characteristics of these diffraction patterns indicated that DNA was formed of two chains that wound in a regular helical structure. In fact, the DNA saga began in 1869. Friedrich Miescher isolated a new substance from the nuclei of white blood cells. Adding alkali made the cell nuclei burst open and releasing their contents, from which Miescher extracted the DNA, which he named nuclein. Analysis of this nuclein showed that it was an acid containing phosphorus, so it do not fit into any of the known groups of biological molecules such as carbohydrates and proteins. Further analysis suggested that chromosomes contain DNA, which led another German researcher, Oskar Hertwig, to declare that nuclein is the substance which is responsible for the transmission of hereditary characteristics. Chromosomes also contain protein, and biochemists were just beginning to appreciate what large complex molecules proteins were. The fragility of DNA was to conceal its underlying complexity for many more years. In 1950, Chargaff pioneered the paper chromatography of nucleic acids, using this to determine how much of each of the component nucleotides was contained in a DNA sample. Each species differed in the amount of adenine, cytosine, guanine and thymine, but within the species, the proportions of each are identical, no matter which tissue the DNA is extracted from. Chargaff's further discovery was that the proportion of A in any DNA molecule, that is adenine in any DNA molecule, was always equal to the proportion of T, that is thymine, and likewise the amount of G, that is guanine, and C, cytosine, always corresponded, a rule that became known as Chargaff's ratios. Please see the figure on screen. This figure shows the representation of Chargaff's rule, that is adenine is always equal to thymine and guanine will be equal to cytosine. Please see the figure on screen. It represents Watson-Crick model of DNA. The features of the Watson-Crick model of DNA deduced from the diffraction patterns are: first one, two helical polynucleotide chains are coiled around a common axis. The chains run in opposite directions. Second one, the sugar phosphate backbones are on the outside and therefore the purine and pyrimidine bases lie on the inside of the helix. Third one, the bases are nearly perpendicular to the helix axis and adjacent bases are separated by 3.4 angstrom. The helical structure repeats every 34 angstrom. So, there are 10 bases, which is equal to 34 angstrom per repeat or 3.4 angstrom per base per turn of helix. There is a rotation of 36 degrees per base, 360 degrees per full turn or 10 bases per turn. Fourth one, the diameter of the helix is 20 angstrom. The orientation of the two strands of DNA is anti-parallel, that is, their 5' to 3' directions are opposite. Please see the figure. This is a figure showing the anti-parallel nature of the DNA. That is, the members of the each base pair can fit together within the double helix only if the two strands of the helix are anti-parallel. A consequence of these base pairing requirements is that each strand of a DNA molecule contains a sequence of nucleotides that is exactly complementary to the nucleotide sequence of its partner strand. The strands are linked by a regular base pairing between the two strands. A, that is adenine, is paired with T, that is thymine, through 2 hydrogen bonds. G, guanine, is paired with cytosine through 3 hydrogen bonds. Please see the figure on screen. This shows the chemistry of complementary base pairing. We can see that thymine binds with adenine with 2 hydrogen bond and guanine binds with cytosine with 3 hydrogen bonds. This base pair complementarity is a consequence of the size, shape and chemical composition of the bases. The presence of thousands of such hydrogen bonds in a DNA molecule contributes greatly to the stability of the double helix. Hydrophobic and van der Waals interactions between the stacked adjacent base pairs also contribute to the stability of the DNA structure.